All right, thank you very much, Lane, and welcome everyone. Good to see that everyone has joined. And I'm going to start sharing my screen. So what I want to do in my part of the presentation is go over a little bit what we will cover in each one of these parts. So already Lane mentioned that we will be talking about data science, we'll be talking about machine learning, and we'll be speaking a lot more in detail about every aspect of these things. Now, the major objective of this data science program and how it is different from some of the other programs that we've been talking about yesterday and we'll talk about tomorrow is that this particular program is designed to give you some more general skills in addition to your ability to analyze biomedical data. So what do I mean by that? First of all, data science in itself is very similar to bioinformatics. So you could define bioinformatics as the intersection of computational tools and methods to organize and manage your data with a focus on biology, whereas data science is a lot more general. So it could be applied to a lot more different applications besides biology itself. And so you can kind of say that bioinformatics combines a lot of the same elements, but with this unique focus on biology. At the same time, when people hear the word bioinformatics, they tend to think of very specific applications of bioinformatics. So people would think about BLAST and kind of sequence alignment and things like that. However, I want to talk today about how the nature of sequencing data is changing, and a lot of the ways that we analyze this data is also changing, bringing to the forefront this need to have data wrangling, data analysis, data visualization, and machine learning skills in what we are able to do. And so I'll talk a little bit more about what we will be able to do in the context of this particular program to help you kind of get those skills. So the program will combine three main modules besides some introductory material. These are the three main modules that you will have to complete: BioML-Logic, BioML-R, and BioML-Python. Now, BioML simply stands for bio data, and ML stands for machine learning. But essentially, it will cover statistical analysis and machine learning that starts from data preparation for statistical analysis all the way to interpretation of what you have been able to identify using a variety of analytical methods. So besides these three modules, there are some additional modules that you can choose to complete. They're not a requirement. So for those of you that have less background in biology, we recommend the course called Bytes and Molecules. Kind of talks a lot about how to apply different types of methods in bioinformatics to different domains, like let's say medical or maybe basic biology. It also introduces a lot of the terminology that you would have to know to orient yourself. So if you come from more of an engineering background, maybe you're not so sure what's the difference between a gene and a mutation, exon and intron, or maybe sequencing DNA as opposed to RNA. And what does that mean in terms of data? Because a lot of the words are used in a different way. So that's something that you can clarify in those introductory modules. And then for some of you that do not have any background in engineering, so you have not done any coding at all, there are some introductory modules like Introduction to Bioinformatics in R and Introduction to Bioinformatics in Python. So those would be useful for anyone that comes without any coding experience. What these modules provide are some exercises that clarify what is the problem or why are we doing what we're doing, how to address this problem from a coding perspective, how to use existing code and maybe modify some of the settings and outputs, and then apply them in an effective way to a data. So here you can see an example from getting started with bioinformatics in R, how to load data, how to visualize it, maybe how to read different types of data, maybe tables or sequences, and how to apply some of these commonly used bioinformatics tools. So this is just to get you started. When you move on to the more advanced courses like Biomedical in Python, for example, you can see that they will use project examples with curated data sets taken from publications. So in other words, a lot of the methods will try to replicate the analysis done in that publication, having to code every step of the way to be able to understand what is the method, what are some of the assumptions about the data, and how can you choose between the different methods to really select the one that is most useful in the context of the research question posed by these different data sets. Now, besides that, we have this parallel. So the exact same thing that you will learn in R, you can learn in Python. Why R and Python? The primary reason is because R has a lot of commonly used packages. You will find typically outputs from different R packages in publications. A lot of the statistical analysis is very easy to use with kind of beautiful visualization right out of the box with minimal modification. So that's why R is very popular in bioinformatics. You have a whole a series of packages being developed every day to address new types of data and new types of analyses. At the same time, in Python, it seems like some of the things are going to be a little bit more challenging in terms of, let's say, visualization and making these beautiful figures that you can just take and use in your publication. But a lot of times, once you get into the syntax, Python tends to be something that people feel much more comfortable with after they've kind of overcome this initial stage of some technical things that you just have to practice and know. So these two languages are very popular, and we'll talk a lot about how to get started, how to set up your environment, and eventually how to use those in the context of a data set that you choose or you pick for your project. Just to kind of give you more in-depth information about how this is actually going to work. So here's an example from the Python version of the course. You can see that there are several different sections here. You essentially will be able to learn the code syntax just by reading about the code. It will explain every single piece of the code. What are we trying to do? Import packages, loading data, removing specific portions of the data, manipulating our data, and then you can practice. So essentially you can replicate this code, run it, and test your results, whether your results are correct or not. And so as you saw here in this little video, you essentially run that piece of code right in the browser, and it will tell you whether your result is correct. Besides that, you will then be able to take this code and modify it so that you understand how to apply it to different types of data sets. So each exercise is going to be first to understand the objectives and strategies, then to learn the syntax or how to code this in a particular language like Python, then how to test your understanding in these coding challenges. And so for the coding challenges, you will get a link to a Jupyter notebook that will typically have some initial code. And then once you look through that code, you'll be given an assignment how to complete it, how to maybe take something that you've learned in the lesson and apply it and write it out in code and make sure that that code works. So a Jupyter notebook looks like this. Essentially, it's like a Google document. In this case, this is a Google Colab, where you can take each portion of your code and run right in the browser. In this example, all of the code is already written for you, but in some cases, you will have to modify or maybe write full pieces of code yourself. And that way you'll learn how not to only learn about the syntax, how to also write it all out yourself. So let's talk a little bit about what the different sections are going to cover. And so this is an overview of all of the topics that we are going to be covering in this program. So we're going to divide it into 3 different parts. The first part is introduction. The second part is methodology, how to understand and apply these methods. And the last section is actually about the application to particular projects. And so the first session is going to give you a sense of omics data, terminology, and key concepts that you just have to know to get started. The second portion will tell you about the details of the methods. What's the difference between different clustering methods, or how to do dimensionality reduction using different techniques for different types of data, and what are the differences between statistical analysis methods and machine learning methods or deep learning methods. And then finally, you will get introduced to a number of different research publications with curated data sets, and you will see how this is used in clinical applications, in biomedical research, and in different aspects of what the industry is doing with clinical trials or with designing new drugs and things like that. So let's talk a little bit more about the different challenges that you should be able to address once you complete this full program. Typically, when people think of big data, they think of data that is intuitive and easy to understand. And in this case, especially in the world of biomedical research, typically that is not true. So there are several different challenges around it. First, this data is unintuitive. So you really have to have a lot of additional information to understand what the data describes. And number two, the data is not structured. So as you can imagine, a lot of these data sets that I have here on the screen come in their original form in an unstructured way. You cannot just take them and analyze them. So the first challenge that you should be able to do after this program is to structure data and prepare it for analysis. What kinds of data are we going to be dealing with? Primarily, we're going to be talking about omics data. This includes phenomics, genomics or DNA variation, epigenomics like bisulfite sequencing, so DNA methylation, or ChIP-seq, where you're looking at histone modifications, and transcriptomic data, including some other types of data sets like metagenomics. So all of these data sets have to be processed to be useful. And we'll briefly touch on how to process data, because essentially sequencing data looks like this. You have short reads. Those short reads have to be aligned to the reference genome. And only after they have been aligned to the right place on the genome, you can actually quantify how many you have and translate this into structured data that is quantified. So you actually have numbers associated with features like genes and samples that represent some biological conditions that we can compare and contrast. To do that, you essentially have to know some of the basic ways that some of these coding languages deal with data. And the way they deal with data, in this example, we're going to look at Python. So in Python, you have two major packages that allow you to work with matrices that essentially represent numeric data that you can manipulate and work with, and pandas that allows you to work with data frames. And so we will learn about how to take this kind of data, prepare it for analysis, load it into the environment that we're working in, for example, R or Python, and then that will make it ready to explore. And so once we've loaded the data, we will start looking at data properties. So how can we estimate different important parameters like distribution, like, for example, different ways of normalizing data to check that the data is kind of consistent with our assumptions about the data, and how to essentially prepare this data for the method that we will use in the analysis, because in a typical way, that method will have some expectations about the data that are important to make this analysis correct and make it interpretable. As a part of that, we will talk about different ways to visualize data. An important part of kind of playing around with the data is being able to, for example, extract different portions of your data frame, combine them together to find a data set that you need. For example, if you're talking about different types of conditions, and those conditions are defined by different sexes, different ages, different disease stages, you might want to look at just some of those. And so one way to do that is you have different types of categorical data, and you want to create a data frame so that you can analyze that, and that's called crosstabs. The other way is you can compare categorical groups. So for example, different age groups, different sexes, different disease types, And then you can create summary of continuous data, for example, gene expression, to visualize it and make it